Right, so let's look at the details of meiosis. Right, we mentioned just a little bit ago in interphase 1, or in interphase, we're going to go through the replication of the genome, of the DNA. And ordinarily, in the nucleus, the DNA is in the format of chromatin. Right, it's loose, it's kind of loose and laying out, kind of like spaghetti strands, and it's open in that way so that, one, if we're going through normal cell processes, we can read the DNA and transcribe it to mRNA and then translate that into proteins. Or in S phase, it's laid out so that way we can make a copy of it, we can duplicate it. Right? Once it's been replicated, as we move into prophase or prophase 1 of meiosis, the chromatin will start to pack itself up and tightly wind itself into the form of being a chromosome. Right? So that's what's happening leading up to the start of meiosis. It's very similar to what happens leading up to the start of mitosis as well. Right? But what we do with these chromosomes in prophase really is very different when we compare mitosis to meiosis. All right, so there are 4 phases we're going to roll through in meiosis 1, and we're familiar with prophase, metaphase, anaphase, and telophase. Right? But because we're going to go through this process twice over, we usually designate prophase 1, metaphase 1, anaphase 1, and telophase 1, this first stage of meiosis. But the key takeaway here is that in meiosis 1, we're going to go from a cell that's diploid in chromosomal number to one that's haploid in chromosomal number. And in humans, we're going to go from 46 chromosomes to 23 in this first major set of processes in meiosis 1. Right, so we're going to hit these along the way. So I'm going to move forward. So this is like an overview image, if you're following along in the presentation, of the steps we're going to see happen in meiosis 1. Right, so let's look at some details about them. In prophase 1, the chromosomes obviously will condense. They'll go from chromatin format to a tightly coiled chromosome form. They'll have their sister chromatid along with them. So they make that traditional X-looking like shape with the sister chromatids attached at their centromeres. Also in prophase 1, though, what makes it different from mitosis is that in meiosis, these homologous chromosomes are going to actually find each other and pair up. And when they do, that means that you're going to have like the paternal chromosome with its sister chromatid in white here, and maybe the maternal chromosome in blue with its sister chromatid, and they're going to find each other and hang out. Right? This process is known as synapsis or synopsis, and the formation of 2 homologous chromosomes close together is known as a tetrad. Tetra meaning 4. And we have 4 chromatids hanging out real close to one another. Because of that, there's an event known as crossing over, where some of the genetic information from one of the telomeres of one chromatid can kind of be switched in position with another one that's nearby. Because if you consider the fact that both the paternal and maternal chromosomes have the similar genes, like the gene for hair color, the gene for eye color, could both be found on the same homologous chromosomes. Whether they come from your mother's side or your father's side of your genetic makeup. So because the coding is very similar in that region, it's easy for, if they get close to one another, to kind of swap or hot swap each other around. So in prophase, we're going to take our homologous chromosomes with their sister chromatids, and we're going to move them in close to one another, and that's going to result in the formation of that tetrad. Now, as most biological processes, it's a little messy, right? We draw it and model it very nice and clean, but when we looked at the cells in our microscope, we could easily see how the dense chromosomes were kind of like almost an entangled mess as they are moved through the different processes. So in meiosis, because we have so many of these chromosomes, the 2 sister chromatids from both so close to one another, their telomeres can actually wrap around one another. Right? And so what this does is cause crossing over. Every crossing over event is known as chiasmata. All right, so this chiasmata event happens. It can happen in multiple locations along a sister chromatid or along a telomere. And what you end up getting at the end then is some variation. Right? You get sister chromatids that are no longer identical because next to its non-sister neighbor, it swaps around some information. Now, there's nothing that says it has to happen. There's nothing that says it has to happen in a certain location. Right? But there are some common regions that seem to have these events happen more often than not. And the odds of it happening are rather high because of how closely packed together these tetrads are as they pair up. All right, so really what we're looking at is that this is going to be this kind of interchange of genetic information that's going to cross between the paternal and maternal genome of that originating organism. And that's going to cause a shuffling of the genetic information, and we term that genetic recombination, sometimes called recombinant DNA. Because we're simply recombining, we're resorting, mixing around all the genes that are present. Still making sure, because this crossing over is happening between homologous chromosomes, we're still making sure each of them has the hair color gene, each of them has the eye color gene, but it's just we've traded places where they're on, or which chromatid are they on. What this does is this leads to a good source of genetic variation. In your overview yesterday, we mentioned 3 ideas as far as what causes genetic uniqueness in our gametes and ultimately in the new individual that's formed through fertilization. And one of those is this crossing over, this genetic recombination that happens during prophase 1. All right, so if we go back to our pop V little guys here, these guys would pair up near one another. I gotta get this guy out of the way. Right, so we have this tetrad, right, and this would be happening with other chromosomes that we have homologous pairs for, right, and they would have crossing over events as well, right, and eventually they're going to make their way to the middle of the cell, so they're going to make their way into the metaphase plate. Keep in mind that after this crossing over happens, some of these chromatid arms are no longer identical to their sister. Right? We still have sister chromatids, but it's possible that, I don't know, if we have some sort of crossing over event happen, that, what was once all red, we have swaps between reds and yellows with the chromatid that are part of the tetrad. Right? So that no longer are, these sister chromatids should be identical. Well, they kind of are because I made them hot swap that way, but they would no longer be identical to each other. So at the end of prophase 1, as we make our way into metaphase 1, we have spindle fibers attaching to parts of the tetrad. The centrioles are making their way to either end of the cell. And instead of individual chromosomes with their sister chromatids being moved around, we have entire tetrads, pairs of homologous chromosomes being moved around. So in metaphase 1, the tetrads are going to be what lines up in the middle. And this is very different from what we see with mitosis. Right? In mitosis, once we have our replicated copies, they would line up in mitosis with their sister chromatid lined up with the centromeres all in one line. Right? So this would be mitosis during metaphase. But in meiosis, we actually have the homologous pairs lining up at the metaphase plate. So the tetrads are lining up. And what this does is it also adds another source of variation in genetics because independent assortment will occur, meaning that because I have these things color-coded on the board or because we haven't color-coded in their illustrations in your note handout, the white and the darker gray chromosomes can be set up to be like, maybe yellow goes to the left, or it could be set up where it's the other way around, that the yellow will be on the right-hand side, right? So that's a possibility. So there's a couple options for these guys lining up at the metaphase plate. And so how many different possibilities are there based on this variation? And as you probably caught in Bozeman notes, or maybe even your flex book, if there's two possibilities, raise the power of the number of pairs that are present. There are 23 homologous chromosome pairs present. That's going to give rise to over 8 million possible combinations as to how these things might be arranged. Okay, so the more pairs you have in place, the higher you raise the number to. Right? Also consider the fact that at the end of meiosis, we're going to end up with 4 gametes that are genetically unique. And only one of them, if we were to actually try to go and push it through fertilization, only one of those 4 can fertilize any given egg. Right? So that even that increases the odds of having an identical individual from the same set of parents. It's even more and more slim to occur, if ever. All right, so whoops, let me go back. Didn't mean to hop that one. There we go. So at metaphase 1, at the end of metaphase, we're lined up on the metaphase plate, and notice we have a tetrad lined up here. All right, that's a different formation than what we see in mitosis. So when you look and compare the two processes, what happens during prophase 1 in meiosis and what happens in metaphase 1 of meiosis really sets the stage to be different than mitosis. All right, now we can also, I guess, again, talk about independent assortment, where in this image, the white is on the left, but on the right side image, the white is on the right. So how we set those guys up will vary the resulting daughter cells that emerge. In your presentation file, I also included another graphic that kind of walks you through the couple possibilities you have on lining up a couple different chromosomes. Remember, in our illustrations, we're working with a diploid number of 4 chromosomes, which means our haploid chromosome count will be 2 chromosomes. All right, so at anaphase, this is going to be where we actually pull the homologous chromosomes away from one another. So the centromeres remain intact. The sister chromatids will be pulled to either side of the cell. And we have 2 chromosomes heading to the left in our image and 2 heading to the right with their sister chromatid attached. What this does then, as it leads into telophase 1, is it's going to end up producing 2 haploid daughter cells, each one with 2 chromosomes contained within. So as we go through telophase, the same steps seem that will happen. The nuclear envelope will reform. The chromosomes will start to untangle back into chromatin. And eventually, as that finishes, cytokinesis will take over, and we will then end up splitting 2 different daughter cells, 2 separate daughter cells. Keep in mind that the chromosomes that we now have in these daughter cells, right, we went from a 4 chromosome parent cell, now we're down to 2 chromosomes in each of our daughters, so we're haploid now. And these haploid chromosomes are genetic recombinants of what we once had, because of crossing over that has occurred. So the uniqueness of these daughter cells has already been somewhat accomplished based upon what happened during prophase 1, and then in metaphase 1 when they split or arrange themselves up. All right, so with these 2 haploid daughter cells, we're going to move directly into meiosis 2. There is not an intermission, there is not an interphase between these steps. We go right into meiosis 2, and since we're going to run through PMAT one more time, we simply say that we're talking about prophase 2, metaphase 2, and so on as we move through the steps. All right, so in prophase 2, this is where it's going to look just like mitosis. So a few of you were mentioning that they're very similar, so I don't know if I didn't include necessarily all the steps in my diagram. This would be where it does line up pretty well with mitosis. Right? The spindle fibers will form. The centrioles in animal cells will show up on either side of the cell and kind of migrate to the ends of the cell. And we will attach those spindle fibers to the centromeres of all of the chromosomes that we have in place. Now, in a human cell, during prophase 2, there will be 23 centromeres, or 23 chromosomes that would be getting attached to spindle fibers at this point. Like in our picture, or in our illustration that we've been working out of, we have 2 chromosomes. All right? So then we move to metaphase 2, and these guys will actually line up in the middle exactly as they do in mitosis. The only difference is we're working with the haploid number. Okay? Ordinarily, like at this point in mitosis, we would have 46 chromosomes lined up in the metaphase plate, but because we're going through meiosis, we have half that. We have 23. In our example we've been working through on the screen, we have 2 chromosomes in each that are lined up at the metaphase plate. Right? If this is very similar to mitosis, then what is the next step and what happens? What's the next step and what happens here? Cogswell, which is known as what phase? We're in metaphase. Anaphase, yeah. So in anaphase 2, just as you would expect, we're going to actually pull the sister chromatids apart from one another now. Remember, these are not really truly sister chromatids anymore. I mean, they're sister chromatids, but they're not identical anymore, right? Because of crossing over that happened in prophase 1. So in anaphase 2, we're going to pull the sister chromatids to either end of these cells. All right, and then in telophase 2 and cytokinesis, we're going to reform the nuclear membrane and also split the cell membrane into total of 4 different cells. So in telophase 2, the nuclear membrane reforms around the chromosomes that are present, the haploid number. They're going to, again, uncoil and uncondense back into chromatin, and we're going to go through cytokinesis. And these 4 cells that have been formed are genetically unique. Now this seems to be a lot of the same that we see in mitosis, just done all for the sake of having 4 unique gamete cells that are haploid in chromosome number. But it's rather important that they have 2 different processes involved with cell division. Because in a multi-celled organism, right, we need mitosis to duplicate, make clones of the specialized cells that we have, the skin cells, the bone cells, muscle cells, brain tissue and cells. And we need those cells to be duplicated for repair and growth. But then as far as procreation is concerned, we need a process that will result in a unique set of genes that when brought together with another haploid cell, form a new unique individual that still has some of the traits and some of the functionalities of the original, but is also able to maybe even have built-in information to adapt to changes in the environment as time moves along. So meiosis is actually a very, very, very efficient manner of getting a unique set of genetic information from that which you already have. All right, so as far as meiosis in male versus female, in males we're looking at spermatogenesis, and in females we're looking at oogenesis. Through oogenesis, you're going to create ova, or the individual term ovum, or the egg cell, and then in males you'll produce obviously sperm. and through spermatogenesis. Now, even though both males and female sexes will have cells that go through meiosis, the timeline through which they happen and the end result is slightly different. So we're going to make mention of those details as we move ahead a little bit. All right, for males and spermatogenesis, this happens from puberty all the way until death. Right, so the male sex is always producing more gametes all the time. The benefit to that, as far as the male versus female sex is concerned, is that environmental effects aren't necessarily permanent when it comes to the gametes. Because if you're always making new gamete cells, then if you accidentally walk through Chernobyl, and you get irradiated, right? Within several months, you'll have a whole new set of gametes for reproduction, right? So what damage that was done when you got irradiated walking through Chernobyl will hopefully not spend too much time there. Will not be lasting necessarily. All right. Also, the gametes that are produced. As we move from the primary spermatocyte to the secondary spermatocyte to finally the haploid sperm cells themselves, they're all viable. Like all 4 gametes that are made in this one process can potentially fertilize an egg. Now, I think Bozeman mentioned this as well. These do not contain miniaturized people in them. All right, it just contains half of the genetic code for a total organism, right? So I think we're beyond that, but it was a rather funny thought. Now with females and oogenesis, this actually happens during embryonic development as well, like before they even are necessarily born. This meiosis process has occurred. So as far as like the viable gametes that a female has, they have their total set already kind of preloaded and ready to go at birth. It's then at puberty then, once every ovulation cycle, a single egg out of that set will be selected to finish maturing and then be presented for possible fertilization. The meiotic process for females, though, we start with our primary oocyte, and then through meiosis 1, we form a secondary oocyte and a polar body. We go through meiosis 2, and then we eventually have an egg, and then a polar body is made, and this polar body from meiosis 1 also goes through cell division, and we end up with 3 polar bodies and 1 ovum. So in the female meiotic process, only 1 out of the 4 gametes is actually viable. The reason? The egg is kind of like the ultimate package. It has half of the genetic information from that individual, again, recombined, so recombinant DNA from the individual in a haploid sense, but it also contains the mitochondria, the organelles, the nutrients, and mechanisms in place necessary for, once fertilization happens, for things to take off. And so this egg cell is it's critically important. I mean, I don't think that, I think that goes without saying. But it also, it's like the full package. It has everything it needs to get things going, minus half the genome. Right? Every one of us in this room, our mitochondria that we have in our body is maternally gifted. Right? We get it from our mothers. It's rather interesting to trace lineage or ancestry back using mitochondrial DNA to do so. In fact, I read an article a week or so ago looking at the Native American trek from, the migration across the land bridge to settle, to have, what we would then term Native Americans inhabit this northern continent. And what's interesting about the research is that they're finding that from the mitochondrial DNA that whoever came over, they can trace their origins back to like 6 to 8 mothers. right? Which is kind of, I mean, if you think about it, that's not too far-fetched of an idea because, I mean, the mitochondria that you have, it was gifted to you from your mom's mitochondria was gifted to her from her mom, and so forth. So it should kind of trace back to a smaller number of individuals, right? Because you're sourcing from mom every time you go back a generation. What happens to these polar bodies that are left over? They'll be metabolized. They'll die. They'll go through apoptosis and they'll be metabolized and recycled or wasted or whatever they need to be. Whatever needs to happen with the components to make them up. So once we look at creating a new individual, right, we need to get these two things together. So we get the egg, we get the sperm, we get those two to deliver their haploid set of genes. And if fertilization occurs, then we term that new individual a zygote, and it'll have a complete set of those 46 chromosomes and move through its growth process, development. I really do think that it's very amazing what what is contributed in the egg gamete in the ovum. The fact that it has as males we contribute half of our genetic information, and that's about it. So I don't know. Feel like maybe we need to step up a bit somehow. I don't know. Anyway, the reason that egg cell is so important and why there's 3 polar bodies and only 1 viable egg is because of what needs to be packaged with that egg cell to function and be viable in its own right. Right, so yeah, sperm provides the haploid set of DNA, and then the egg provides everything else. All right, so if you wanted to compare them to each other, mitosis and meiosis, right? In mitosis, we're going to produce 2 genetically identical cells. We mentioned that several times already. In meiosis, you have 4 genetically unique gametes. Mitosis is going to occur in somatic cells versus meiosis occurring in sex cells located in the ovaries of female and the testes of male, for the human species anyway. And then DNA is going to replicate once and divide once in mitosis. DNA will be replicated once in S phase in meiosis, but then we go through 2 cell divisions. And finally, in mitosis, there's not crossing over, which is a good thing, because if we just want to make another skin cell or another muscle cell or what have you, we do not need to be recombining the DNA to do that. We want an identical cell because it seems to be functioning okay already. We don't need to rehash anything at this point. And then meiosis, we're going to cross over during prophase 1, and that's going to lead to genetic variation. Okay. I did include just a summary illustration of the differences between mitosis and meiosis. You can kind of look that over at your leisure. But the last thing to mention today is what happens when something goes wrong with meiosis. Just like with mitosis, we can have things go wrong regulatory-wise in the cell cycle in general, or in mitosis going through unregulated cell division. We also have issues that can occur during meiosis because it's also a complex process. Right? Non-disjunction is probably the most common issue with meiosis. And what happens is a pair of those homologous chromosomes do not get separated during either anaphase 1 or anaphase 2. Because we line them up at metaphase, and then at anaphase, remember there's usually a checkpoint that says, all right, the tension on the spindle fibers is equal, and we're ready to haul things across to either side of the cell. Something goes wrong there, and we don't haul them across the cell. And so what will end up happening is you get gametes that have an extra set of chromosomes, or those that have none at all of that chromosome. Now, if this happens a lot, I mean, it's pretty obvious that if you're missing 2, 3, or 4 chromosomes, that gamete is probably not going to be kept. It's going to go, or it may result, if it is kept, if fertilization were to happen, it won't continue. It'll go through apoptosis because something's very severe is wrong. But we do have cases where this non-disjunction does end up causing a viable embryo. All right, so this graphic organizer is kind of showing you what happens when non-disjunction happens during meiosis 1 and when it happens during meiosis 2. Notice that if we do not separate the tetrad in meiosis 1, then we end up with a daughter cell that has an extra chromosome in it and one that's lacking that chromosome altogether. If we look at what the result is for gametes after meiosis 2, every single one of the gametes has a problem. Either they have an extra chromosome, so n+1, or they're short one, n-1, whatever the haploid number might be. If that happens in meiosis 2, then we have half of those gametes are fine. The other half, one is +1 and one is -1, the n number. All right, so regardless if it happens in meiosis 1 or 2, the problems with the gametes is still the same. Either you have an extra or you're short one. All right, some common disorders that are associated with non-disjunction: Down syndrome, Turner syndrome, Klinefelter syndrome, and even Edwards syndrome are due to non-disjunction. The names, the official scientific names of them, kind of give away what the karyotype looks like. Right, like trisomy 21 means 3 of chromosome 21. So those people who suffer from Down syndrome or live with Down syndrome have 3 chromosome 21s, right, which causes some developmental problems and associated with some stature, usually shorter in stature. But again, I think we probably know many individuals that have Down syndrome that are absolutely have fully functioning and fulfilling lives, and are super people and individuals in their own right. So it is a non-disjunction disorder, but it does not mean it's fatal. Right? There's some quality of life there. Turner syndrome is when you have only 1 sex chromosome. Trisomy 23 is when you have 3 sex chromosomes, and then trisomy 18 is when you have 3 chromosomes.